Okay, it is an honor to be invited here to share my recent work with all you. Meanwhile, allow me to extend my sincere thanks to conference group. Today, I will present about the theme about the autophagic pulse cell cycle rest to modulate stress responses. My name is Pan. I'm from the Northwest Agricultural State Universities. So, allow me to introduce my university about the Northwest Agricultural State University. So, our journey began in 1934. We are proud to be located in Yangli, Shanxi Province, a place where ancient agricultural traditions meet technologies. This ubiquity advantages offers our students and researchers a perfect place environment for learning and innovation. To hear exciting that we are close to the terracotta armies, the most attractive and historical place. And besides, you can have a look at the Ding in Chinese names from the West Zhou dynasties and the gold bowl from the Tang dynasty in Shanxi historical museums. So, welcome to Northwest Agriculture, the first university. So, now, let me introduce the background of my themes. As we all know, abiotic stress is widely recognized as a major limiter to plant growth, development, and crop production. So, the extremely temperature drought, salt stress, and heavy mental challenges caused a sharp increase in reactant oxygen species throughout, with finally leading to the damages. So, to cope with these difficult conditions, plants has developed a series of evolutionary conserved stress responses mechanisms. Among them, the damage response DDR, cell cycle arrests, and autophagy are striking control pathways. More studies show that the damage response, cell cycles arrest, and autophagy at the same time could be activated together. And these three proteins do not work alone. They are close connectors and work together to help plants resist stress. So, but the exact molecular links between these pathways in plants is still not clear. In particular, how autophagy controls nucleus damage signals and cell cycle growth is an important question that needs to be more researched. We first focus on the key control system of the plant cell cycle. As well known, cyclin-dependent kinases CDKs are the main drivers of the cell cycle's progress. Their activities by cyclins CVCs and the phosphatase CTC25, and inhibitors by CDK inhibitors and the kinase V1. However, plants have its unique control ways. They do not have many and proteins like CIP, CDK inhibitors, and the plant V1 does not directly reduce CDK activity through phosphorylation. So this brings up questions about how exactly do ATL V1 regulate a cell cycle in plants. So, by screening for suppressor of the genes in ATR mutant plants, we found a group of key proteins named SALT1, SALT3, and SALT4. Most we call them salts. Using genetic materials, we build a signal pathway centered on the ATR V1 axis. These pathways combine salt, SOC1, CAS, and cyclins to work together and control cell cycles arrest caused by stress. Specifically, the F-box proteins FBR17 act as IE3 evocating agas. It mediates the CDKs inhibits KP6 and 7 for degradations. Next, in order to identify the new compounds of DDR, we performed a large-scale forward genetic screen for DDR mutant. One of these mutants, called DDR7, is more resistant to inflammation, which causes DDA double-strand breaking DSV. So we used the EDO to label the newly made DNA. As you can see in Y-type plants, the number of EDO labeled cells dropped significantly after inflammation treatment because of the cell cycle's arrests. However, this drop was much weaker in DDR7 mutant, which means ATG5 helps promote cell cycle's arrest. DDR7 codes for the autophagy-related proteins ATG5. So, as we all know, the autophagy happens in the cytoplasm, where DD damages takes place in the nucleus. So we wonder how a protein in the cytoplasm controls the DNA damage response in the nucleus. So we examined the subcellular localization of ATG5 proteins before and after the inflammation treatment. Interesting, we found that the ATG5 proteins translocated into nucleus after inflammation treatment. Then we want to know the focus of DDR of the ATG5. So to investigate its function, we first tested whether it maintains its kinetical autophagy role and the damages using ATG8 as an autophagy marker. We found that inflammation treatment at the same time caused ATG5 to move into the nucleus and strong activities autophagy. This finding demonstrated that ATG5 modulated the DNA damage response, so both autophagy dependent and independent mechanism. With the created transgenic plants to prove it, we created the transgenic plants expressing nucleus and cytoplasmic restrict ATG5 and found that both types of transgenic plants partially restore the normal DNA damage responses in the ATG5 mutant. This results further prove that ATG5 controls the DNA damage response, so both autophagy dependent and independent mechanism. So next, we want to further improve the processes nucleus function of ATG5. We adopt the immunoprotection combined with mass spectrometers, IPMS, to scrub its potential binding proteins. This data analysis identified KP6 as a candidate protein that interacted with ATG5. When they used the interactions through several biochemical tests. Among these improperties, KP experience confirmed that inflammation experience treatment significantly enhanced the physical interactions between ATG5 and KP6. And such enhanced interactions probably attribute to the nuclear ATG5, which bring it close to target proteins inside the nucleus. And furthermore, we demonstrate that the nucleus localization ATG5 effectively inhibits FBR17 mediated degradations of KP6, thereby stabilizing the skin cell cycles inhibitors. Collectively, these findings lead us to hypersensitivity ATG5. Positivities regulate Cap6 accumulations to enforce cell cycle's arrest. Next, to verify this acceptance, we overexpressed Cap6 in the ATG5's mutant backgrounds. Notably, Cap6 or expressing in the ATI mutant rescues the insensitive phenotype of ATG5, thereby supports our hypothesis. We next investigate how ADN damage checks ATG5 nucleus translocations. IPMS first identifies GF3 as a candidate interacting partner of ATG5 and the mechanisms. Co-immunoproteasase assays confirmed these actions and the interactions and the bonding interactions significant attenuated following inflammation treatment. And when GF3 was expressed in GI backgrounds, more ATG5 was retained in the cytoplasm. Taken together, this result suggests that GF3 combines with ATG5 and keeps it in the cytoplasm at no mechanisms. When genotoxic stress the disruptions of the ATG5 and GF complex, free ATG5 and enables its movement into the nucleus. Notably, GF3 belong to the connectors 43 protein families with specifically recognizes and bonding phosphorylated motifs. So this character implies that stress-induced separation between ATG5 and GF3 is likely scarred by dynamic phosphorylation events. So to clarify the genetic relation between the GF3 and ATG5, we generated two independent GF3 mutant alleles. Both alleles except pronounced hypersensitive to inflammation treatment. Strikingly, the ATG5 GF3 double mutant displays insensitive to inflammation treatment, effectively suppressing the hypersensitive phenotypes of the GF3's single mutants. So this genetic analysis confirmed ATG5 as the downstream of the GF3, indicating the GF3's negativities regulates ATG5 activities. So based on this genetic regulatory relationship, we first explores the molecular mechanism caused by the disruption of ATG5, GF3 complex and stress. Since GF3 typically recognized phosphorylated motifs, we test whether ATG5 undergoes stress-induced phosphorylation. Notably, ATG5 exhibited no detectable phosphorylations under the genotoxic conditions. This finding directs us mechanism focused toward the bonding partner itself, leading us to hypothesize that GF3 rather than ATG5 may serve as the directors phosphorylations. To test this hypothesis, we performed in vivo phosphorylation assays and demonstrated that the inflammation treatment induced GF3 phosphorylation, and S14H and S14235 are the modification shots. And two next to identify the upstream kinase responsible for GF3's phosphorylation, we scrubbed the kinase compounds and found that the contain direct phosphorylase GF3s and consists with from the expressions of a non-phosphorylatable form of GF3's complete block to the nucleus translocation of ATG5 upon the damages. This results indicating that contain mediated phosphorylation of GF3 served as a critical switch for ATG5 nucleus import. plants contain focusing as a catalytic alpha subunit of energy accessing complex with its nucleus activated by the upstream SNK kinase. However, the functional connectors between the SNK and contain signalings more moderate, and the DNA damage response remains largely uncharacteristic. So, we further verify the relationship between the contain and GF3 cells genetics analysis. So, as we can see, the mutant sold tolerance to blemish treatment, and showing similar trait to the ATG5 mutants, and the GF3 contain double mutant has much slower root growth than the contain single mutant, suggesting that GF3 works downstream of the contain, and after conferring the regulated pathways, we further explored the function of contain and its upstream activities as in DNA damage response. So we then explore the upstream regulators of contain, regulate and reduce so that the blemish treatment triggers contains phosphorylation, and the phosphorylation straight rely on SNK, the upstream. GF3's phosphorylation need both the SNK and contain to exist. These findings collectively confirm that SNK acts as a positive upstream regulator of contain and drives the phosphorylation cascades essential for an effective DNA damage response. After clarifying the downstream phosphorylation process, we further study and why the SAK serve as the top kinase in this pathway. Meanwhile, it remains unclear how SAK and gas activities are regulated and genotoxic stress. To resolve these issues, we analyze the subcellular distribution changes of SNK blemish treatment from the nucleus to the cytoplasm and accompanied by the upper vessels SNK phosphorylation. And this phosphorylation event job us to search for its upstream kinase. Since ATM is widely known as a causes in DNA damage response, we proposed that ATM could directly phosphorylate SNK. We thought that ATM not only bonds to and phosphorylate SNK, but also straight controls its nucleus cytoplasm cycle in the response to the damage. So, based on this above finding, we found a comprehensive molecular pathway linking autophagy coupled cell cycles arrest to regulating gene damage response. Upon gene damages induced by stress in plants, ATM its activities and promotes the phosphorylation and nuclear export of SNK as phosphorylated SKS activity contain within phosphorylase GF3. Phosphorylated GF3 is separated from ATG5, and the liberated ATG5 translated into a nucleus. Nucleus ATG5 suppressed FPR70 mediated degradation or ATG KP6 step by lysine KP and inhibits CTK activities. Ultimately striking cell cycle arrest. Meanwhile, ATG5 participates in autophagy to control the elements, proteins, and organelles, thereby enhance plant stress tolerance. And notably, this regulatory mechanism is evolutionarily conserved in rats. As you can see, or ATG5 undergo nucleus translocations in response to damages, and the RAS OS ATG5 mutants exhibited hypersensitivity gene damage, and in human cells, humans ATG5 repressors SKB2 dependent degradation of B27. The result demonstrated that ATG5 mediated cell cycle regulator is highly conserved across plants and human cells. Okay, today I just briefly outline the key findings of this test. Feel free to discuss it with me if you are interested. You can send my emails. I sincerely appreciate my postdoctoral supervisor and the other co-author for this contribution to this week.